again for the introduction, and thank you guys all for joining me this afternoon just to talk a little bit about wildlife rehabilitation, which is one of my favorite topics. So I'm going to start the talk this afternoon talking about just like an overview of wildlife rehabilitation, some of the principles and goals underlying wildlife rehabilitation efforts. And then I'm going to tell you a little bit about my research. Actually, a good portion of my research is based on rehabilitating animals, especially sea turtles, and so I'm going to tell you about that work. So some definitions for you. Rehabilitation means to restore to customary activity through therapy and education, and that usually includes treatment, which means to give medical aid to. One of the main concepts of rehabilitation is that the focus is on the individual animal or human rather than the population at large. There's a lot of underlying ethics surrounding wildlife rehabilitation that I think are important to consider. When we look at animals and animal welfare and animal treatment, animal medicine, a lot of times it comes down to decisions that are made by the animal's owners. And with our pets, we know who the owners are. Farmers, we know who the owners are. But who owns wildlife? So legally, wildlife is owned by all of us. It's owned by the public trust, and that's in the United States. So we all have a stake in animal welfare when it comes to wildlife. And I think most people would agree that we have a moral imperative to humanely care for a wild animal in distress as their owners, and especially when the distress is due to human activities. So I'll just give you some examples of some horrible things that have happened to wildlife. This is a harbor seal who has outgrown his packing strap entanglement. This is a oiled sea turtle. This is from the Deepwater Horizon oil spill in the Gulf. Here is a Florida manatee who succumbed to respiratory distress because of harmful algal blooms that are ultimately due to human activities. This is a sea turtle in rehabilitation at the Houston Zoo who swallowed some fishing hooks. Here's a California sea lion who some nasty person shot with a bow and arrow, probably because either for fun or because the animal was raiding his fishing lines or nets. This is an American bald eagle that has lead toxicosis from scavenging on an animal carcass that was killed with lead bullets. And this is a patient that was at the International Bird Rescue Center in California. This pelican, they think a fisherman he was probably, again, raiding the line or the net, and the fisherman got mad, caught up the bird, and cut his pouch with a knife, which is a horrible thing to do. But actually, this is a good story, and this one, too. These animals were rehabilitated and released. So just some examples. So when we talk about the principles of wildlife rehab, programs to rescue and rehabilitate sick animals are founded upon two general principles: altruistic assistance to the sick or injured, and conservation of wild populations. And in the process, we gain medical and scientific knowledge and provide a valuable interface between wild animal welfare and the public. The ultimate goal of wildlife rehabilitation is a successful transition and return of an individual animal back to its natural habitat in a fit and healthy state to reproduce and perform all natural functions and behaviors benefiting the environment and the species. So I think that's important to remember, that the outcome is release. Conservation, so the second principle of wildlife rehab is conservation, as I just mentioned. Conservation is a realistic motive for reintroduction programs for threatened species. So yes, reintroduction programs for threatened species such as a rhino. For robust populations, however, conservation is not a defensible argument for rehabilitating animals, because reintroducing a few or even a few hundred of these animals will be of little benefit to most stocks and even less benefit to the population as a whole. So when we talk about rehab, a lot of people want to focus on the good things, but there are also a lot of downsides to rehab that a lot of biologists will argue. First of all, it's extremely expensive. There are several recent studies indicating that rehabilitation of a single animal can cost over $100,000, depending on the species and the duration of rehabilitation. And this cost may be justified for endangered species with populations that are imperiled. However, the costs are very difficult to justify for non-threatened animals. Also, many biologists would argue that rehab facilities divert money away from programs based on wild population conservation and habitat protection. Rehabilitation success rates vary by species, but they tend to be quite low. For example, the sea turtle rehabilitation success rate in Florida between 1986 and 2004 was 36.8%. And when we say success, that means the animal was released. In another study in California, 14% of 35 rehabilitated dolphins over a 25-year period were released. So generally low success rates. There's a few long-term follow-up data on rehabilitating animals. The data is logistically difficult to obtain and maintain. It's expensive, and it's difficult to track animals after you've released them. That's the bottom line. Some people argue that wildlife rehabilitation has no benefit to the population unless the animal reaches sexual maturity and reproduces. Minimally, the animal should have an annual survival expectation equal to the age class of that population, and anything less than that is arguably a postponement of the animal's fate by the number of days that it was in a rehabilitation facility. Another concept is that rehabilitated animals may be returned back to health, but it doesn't necessarily mean that they're releasable. The animal in order to be released, a wild animal has to be able to perform all of its natural functions, including movement, maintaining stable body weight by normal foraging behaviors, have normal blood health, have no indication of infectious diseases, and not be habituated to or dependent upon humans for survival. So there's some pretty tall criteria, and the fact is that a lot of rehabilitated wildlife species end up as permanent captives. There's also a risk of pathogen spread. So when you bring an animal into captivity, there's a risk to the humans who are interfacing with that animal being exposed to zoonotic diseases. There's also a risk when you reintroduce that animal into their wild population that they may have picked up some diseases during captivity that they would then take back to their population. So those are the downsides. There are a couple other ethical dilemmas that underlie wildlife rehabilitation. There's a lot of research that's been done on these concepts. So the main question is that, do we invariably act in the best interest of the animal, or are our best interest, or our number one focus is on like our altruism and that it makes us feel good to rehabilitate the animals? Also, the question is it humane to rehabilitate a non-releasable wild animal? And also, how can we ensure a released animal will survive in the wild or contribute to the population? Like I said, you need long-term follow-up data that can be expensive to do and difficult to maintain. There's also a rehab bias, and I just thought this was worth mentioning. We tend to focus on species that we have a special affinity towards. There's this cute and cuddly bias. We like adorable animals, and who doesn't? There's also a flagship species bias where we tend to gravitate towards charismatic megafauna and kind of ignore the less charismatic species like snakes and things like that that people may not relate to as easily. So the truth about wildlife is that humane rapid euthanasia is often the best option for injured or sick wildlife. Euthanasia stems from the Greek word euthanatos, which means good or easy death. And the key to ensuring the welfare of individual wildlife casualties is a proven process of triage that enables rapid euthanasia of animals that are considered unlikely to be released back into the wild. Success in this sense may be judged in terms of preventing unnecessary suffering, whereby a high level of euthanasia could be considered successful. Once a decision is made to bring the animal into rehabilitation, success is then defined as bringing an animal back to health and releasing it into its natural environment, and benefiting from this process in terms of public education and scientific knowledge. But either way, the triage process should be done quickly, animal side, and the decision should be made quickly if you are going to euthanize. So again, the key to successful rehab is a well-defined triage criteria, which is supported by research, effective, appropriate intake criteria, which is often species-specific. You need highly trained personnel and lots of infrastructure, including facilities, funds, and flexibility, and usually a lot of those things. So those are just some of the overall concepts about rehab, and now I'm going to talk about my favorite part about rehab, which is access. So an important benefit of animals cared for in rehabilitation is their role as sentinels of ecosystem health. Because rehabilitating wild animals are so accessible, they can help contribute to the developing reservoir of information on care and medicine that we can then apply to free-ranging wildlife needing help. And this health knowledge is crucial for wildlife conservation, and also veterinarians gain skills and knowledge from rehabilitating animals that enable them to play a vital role in conservation and also in turn to rehabilitate animals in the future better. So some of the things that we learn from access to rehabilitating wild animals is information on physiology, life history, nutrition, behavior, diseases, toxicology, and medicine, including treatment and diagnostics. Wildlife rehabilitation centers can be a valuable resource for the surveillance and monitoring of pathogen activity in wildlife, which in free-ranging animals can be cost prohibitive and logistically challenging. So although there are some inherent biases in this approach, obviously, it can still provide valuable information to assist in management and conservation decision-making, including approximations of disease prevalence in the general population, species variation in disease susceptibility, clinical syndromes, standardization of diagnostic protocols, development of medical approaches, therapeutics, and outcomes associated with various pathogens. And we can use this information to make these population-wide inferences that we can apply to general wildlife populations, and that we can also then, in turn, apply back to rehabilitating wildlife to inform and improve future efforts. Okay, so that's just my introduction, and now I'm going to tell you a little bit about my research. For the past 7 years, I've been working on figuring out this disease that affects sea turtles called fibropapillomatosis, or FP. And since you live here in Florida, you may be familiar with it. It's a debilitating infectious neoplastic disease that affects all sea turtle species but has reached epizootic status in green turtles, meaning it's an epidemic in many green sea turtle populations across the world. It's been reported in all oceans in many countries. It tends to mainly affect juvenile green turtles after they migrate back to their coastal habitats, so usually around 4 to 7 years old. It can affect they can have tumors on any part of their body, including internal organs, and it can be fatal in some cases. There's a virus associated with these tumors called chelonid herpes virus 5. It's the presumed etiologic agent of FP. Here's the virus right here. I say presumed because like many herpes viruses, this virus is not able to be isolated in cell culture. However, the tumors have been proven to be transmissible via cell-free tumor extract, meaning that a researcher cut off a tumor, ground it up, passed it through a virus-sized filter, injected it into naive turtles, and they grew tumors. This virus is consistently associated with FP tumors via multiple molecular techniques. Herpes viruses group into alpha, beta, and gamma herpes viridae, And this one is here. It groups with the alpha herpes viruses. Phylogenetic studies show that the virus is co-evolved with its sea turtle host, meaning that the recent epizootic or epidemic is probably not related to the virus, but probably something related more to the host, which is the turtle, or its environment. Other factors that have been associated with FP, most significantly is the nearshore shallow water embayment, so they tend to get tumors when they move from offshore to inshore. It's also been associated with pollution, eutrophication, harmful algal blooms, and invasive forage species like macroalgae. It's been associated with warm water temperatures, which is not that surprising because a lot of herpes viruses are temperature sensitive, and also with host immunosuppression. So the reason that I'm interested in FP is because when we talk about emerging infectious diseases these days, generally there's this like a triad that involves the pathogen, the host, and the environment. So usually there's a bit of environmental degradation or contamination or both. There's also host immunosuppression, and then you have a pathogen, and when these three things meet, we see diseases that may not have been a problem 100 years ago. Again, you have environmental contamination or degradation, including exposure to this type of environment. Then the host is immunosuppressed, probably related to the environment. So they have decreased cell counts, decreased cell function, and then they have an increased susceptibility to pathogens that normally wouldn't affect them in that way. And in some cases, you get neoplasia, which is cancer. So that's what we're seeing with this disease. And you can see that, again, it fits in really nicely with this paradigm. So we have the host, which is immunosuppressed, probably due to a degraded environment. And then we have a pathogen, which historically didn't cause disease, and now it does. In rehabilitating turtles, FP is a huge problem. It's infectious. It's horrific for the individual animal. It tends to necessitate special husbandry conditions, including quarantine, like stringent quarantine. The tumors can influence clinical progression, extend rehabilitation duration, and complicate prognoses. And here's some examples. This turtle came in, I think she was hit by a boat. This is at the Georgia Sea Turtle Center. And she was in rehab for over a year. And she was doing fine, getting ready to be released. She was quite robust. And then the staff noticed this bulge in her plastron. They did an MRI, and it turns out she had a huge internal tumor, and she was euthanized. This is another turtle who came in with a boat strike wound on the carapace and was progressing quite nicely, and then developed these tumors within the boat strike lesion, and the turtle was euthanized. So I got interested in around 2010 during an externship. When I was in vet school at the Georgia Sea Turtle Center, I had some patients. Some of these turtles I just showed you were some of my patients, and I became really interested in this disease. And so I started out my journey of this research doing a retrospective case series analysis of all the turtles that had or developed FP tumors at the Georgia Sea Turtle Center since it opened. So it's over a 5-year period. And we looked at the occurrence, clinical presentation, and outcomes of disease. There were 25 turtles overall, which represented 12.6% of all the green turtle patients at the Georgia Sea Turtle Center, which is not insignificant. In sea turtles, we age them by looking at their size. Generally, it's called the straight carapace length. So the bigger they are, the older they are. And you can see that it's pretty consistent that the juvenile turtles, these smaller animals, are the ones who are most affected by the disease. In this study, we also developed a modified tumor scoring system based on tumor number, size, location, and severity, so like how aggressive the tumors are. And the tumor scoring system ranges from 0, which is unaffected, 1, mildly, 2, moderately, and 3, severely affected. We found that 48% of these turtles developed tumors over 1 week after entering rehab, and that 10 of them did so during the warmer months. So again, there's a temperature component to this, to the tumor outbreak, and also probably, so they developed tumors after they entered rehab. So they were either infected when they entered rehab or they became infected in rehab. And what I think is most likely is that they were already infected, and the stress of whatever brought them into rehab and the stress of being in captivity led to them developing tumors. FP tumors were most often first observed in the inguinal regions here, so we can advise rehabilitators to focus on these areas, looking for new lesions. And we came up with this estimation that overall about 10% of rehabilitating green turtles may develop FP within 90 to 120 days of rehab, which hadn't been described before. We found that ocular tumors, these eye tumors, were observed in about 2/5 of the tumored turtles. And turtles with ocular tumors were over 8 times less likely to survive rehab than tumored turtles without ocular tumors. So basically having tumors in your eyes is bad. And we also found that 10 out of 25 of the turtles had only these plaques, these flat plaques, which histologically and molecularly were consistent with FP. But these turtles were significantly more likely to survive rehab, and over 50% of them had spontaneous tumor regression, whereas none of the other turtles that had the other types of tumors had spontaneous tumor regression. So if you have flat plaque-like tumors, it's good. Laser surgery is the treatment of choice for tumor removal. And we found in this study that the number of surgeries was not significantly related to case outcomes. So a turtle could have up to 6 tumor removal surgeries, and it wouldn't necessarily affect the outcome of the case. So here's a good example. This turtle had severe FP. This is a patient at the Marathon Sea Turtle Hospital. You can see she was blind. This is a pretty nasty case. So here she is having her tumor removal surgery, and then this is the same animal. So just because they look horrible doesn't necessarily mean that they're going to have a bad outcome. I think that's important to note. However, we also found in that study that 38.5% of turtles that had their tumors removed experienced tumor regrowth within 36 days. So here's a turtle. These are the little FP lesions just developing in the inguinal regions. This is the day that they were removed, right after surgery. And then 15 days later, you can see the tumors are back, and they're even worse than before. And this fits in with what we know about herpes biology, right? So herpes is for life, as we all know. And just because you remove the tumors, you're not removing the virus. And so that's why it's so important to bring the health of the turtle up as well as removing the tumors, because the turtles, their immune system has to be able to combat the virus as well. Because of this study, this little study, we came up with a lot of recommendations for rehabilitators, including for green turtles, trying to release them within 90 days, if at all possible, to avoid tumor development in captivity. Because once they develop the tumors, then you're in for the long haul. The animal has to be able to survive in its current condition, like I told you guys before. But we also came up with the recommendation that mild, stable, cutaneous FP that's not progressing, and the animal's otherwise healthy, eating on its own, healthy blood values, they can still be candidates for release. And this wasn't recommended before. But because of what we know about herpes virus, we know that they're not going to get rid of that disease. And the most important thing is that the turtle overall is healthy. So not just because they have tumors doesn't necessarily mean they can't be released. We recommend that they're released where they're found to avoid introduction of new disease into a naive population. Water temperature over 18 degrees is preferred. And if possible, it's important to track the patients, just like I told you before. We did the same thing for loggerhead sea turtles in the southeastern United States. I looked at 3 rehab facilities. Just basically, briefly, we found that FP is not really a problem for loggerheads. They do get tumors, but they're typically incidental or mild. They are usually not that aggressive. Interestingly, they get these oral tumors that green turtles in this area don't really get. They get a lot of ocular tumors, but they're usually mild. And exposure to green turtles is a risk factor for loggerheads in captivity. So we recommend that green turtles are kept quarantined away from all other species, and then green turtles with FP are kept in an even higher level of quarantine away from all other green turtles as well. However, we did find in this study that for individual loggerheads, FP can be a huge problem, just kind of like how diseases in people could be. So we had this one patient who had severe FP. She was really sick. This is from her intestinal tract. She was euthanized. And, but we actually learned a lot about this turtle, and so far there have been 3 studies, and I'm involved in a 4th just on this one turtle alone. So she provided us with a lot of information, even though she died. So ongoing research, what I'm doing now to follow up with this is I'm doing it again. I'm doing a case series analysis, but on a much bigger scale, and including all species, not just green sea turtles, involving the Marathon Turtle Hospital, the Georgia Sea Turtle Center, the Whitney Lab, the Brevard Zoo, and the Clearwater Marine Aquarium, hopefully. And we think we'll have hundreds of cases in this study, so hopefully we'll get a lot more of this type of information that can then be passed on to rehabilitators. And the questions that we're asking are, does FP tumor score accurately predict rehabilitation outcome? What % of surgically removed tumors regrow? In what time frame do the regrowths occur? And is there a seasonality? So again, looking at temperature. So changing gears, I did a follow-up study of rehabilitating green sea turtles using molecular biology to try to get a better idea of the herpes virus itself. For this study, I developed, validated, and optimized a PCR assay. PCR stands for polymerase chain reaction. It's just a way for us to detect and quantify pathogen DNA. In this method, we can provide absolute quantification of viral DNA. The assay is extremely specific, it's very sensitive, and it's repeatable and robust. And using this assay, I evaluated the relationship between chelonid herpes virus DNA loads and clinical disease in rehabilitating turtles. So we looked at 351 samples taken from 67 turtles, 23 with FP tumors and 44 without FP tumors. We analyzed their blood, their plasma, cloacal and oral epithelial swabs, feces, urine, and skin biopsies. And this is the data that we got. We found, so red is turtles with FP and blue is turtles without FP. And then these are the sample types where we got some results. If the sample types aren't mentioned, that's because none of them were positive. Interestingly, we found viral DNA in urine samples, which has never been described before. We found viral DNA in blood, which has never been described before for this disease. In cloacal swabs, again, a novel finding. And we found it in a lot of skin biopsies, so, which is something that we had found with previous studies, but it just supported the data. And you can see that turtles with FP had a lot more samples that were positive, but it was pretty interesting that we had these asymptomatic animals that also had positive samples. So basically telling us we have asymptomatic infections in some green turtles. When we look at the level of agreement between the different sample types taken from an individual turtle, I thought it was really interesting that there was 100% agreement between blood and urine taken from the same turtle. We know that urine is a filtrate of the blood, so this points to the potential that urine is a method of transmission of this virus. The fact that we found it in normal skin in this and another study of free-ranging turtles points to normal skin being a potential route of transmission as well. And from non-tumored animals as well as from tumored animals. I followed up this study by taking extensive samples from 10 green sea turtles at necropsy. 5 of them had tumors, 5 did not. We took samples of all of their major organs, their neurological tissues, including optic nerves, nerves innervating their front flippers, and nerves innervating their hind flippers, skin, tumors, blood, and urine. These are some of the animals, just to give you a picture. And this is a tumor that was on the kidney of one of the animals. This is the tumor on cut surface. You can see the kidney was still trying to make urine, and we took samples of this fluid and did PCR on that as well for viral DNA. Interesting results. We found, again, the urine samples were positive for viral DNA. And then this was really surprising, or to me, it was surprising that a lot of the kidney samples were positive for viral DNA. And also some of the nerve samples were positive for viral DNA, which has never been described before. We know that alpha herpes viruses tend to go latent within nerve cells, and so this pointed to a potential site of latency, which has not been described for this virus. So there's a lot of stuff that we don't know about this virus, and I think by understanding the basic biology of the virus, then we can understand more about the disease process. So this study gave me more questions than answers, but basically made me wonder if the kidneys are a site of persistent viral infection, probably related to the presence of the viral DNA in urine, pointing to a renal urinary route of viral excretion. Also, the presence of the viral DNA in nerves points to, again, potentially a site of latency of this virus, which is typical of this type of virus. And the sites where we found virus-positive nerves were nerve cells that innervated parts of the body that had tumors. So we found an ocular nerve, or an optic nerve, that was positive for the virus in a turtle that had a tumor in that eye. We found brachial plexus nerves that were positive for the virus in turtles that had tumors in those flippers, and so it supports that idea. So when we put all this information together, we can kind of get a sense of this clonid herpes virus infection cycle, whereby the virus, this is our little virus, enters the skin, enters the host. We're not sure of all the transmission routes at this point. The virus undergoes primary replication and then disseminates throughout the body via blood and or nerves, probably to the kidneys and potentially other sites of persistent secondary infection. The virus probably establishes latency, maybe in nerves, maybe in other things. The virus is then shed. We know the virus is shed through the skin and through tumors, and maybe through the urine as well. And then to follow up on this research, now I'm doing a study with University of Florida and the Brevard Zoo, where we're looking at abnormal kidney samples from green sea turtles. So you can see this top turtle, this is a normal kidney sample here, and then this is in situ, or in the turtle. And then this is an abnormal kidney. We have generalized severe fibrosis tracked through the medullary regions. And this lesion is extremely common in juvenile green turtles in Florida, to the point where it has been called normal because it's so common. And we're wondering if maybe it's due to herpes virus infection. And so for this study, what we're doing is, again, molecular biology paired with histology to see if maybe these types of lesions test positive for the clonid herpes virus, and maybe that's why we're seeing those lesions even though these turtles don't have tumors. And throughout this research, we found a lot of subclinical disease carriers. So you guys may not realize it, but subclinical disease carriers are really important within the epidemiology of a disease. So the fact that we have animals that are infected but asymptomatic points to an environmental and or host factor that plays a role in tumor development, like I said before. It also suggests that this virus is endemic in some populations. These animals serve as a reservoir. So you can see in this diagram, these are the subclinical infected animals. They serve as a reservoir that can lead to infections in susceptible animals, just as, to some extent as much as clinical animals can serve as a source of infection. And so they can be a source of viral dissemination within a population. And this actually probably explains why there was one study where they looked at the viral exposure of this virus in green turtles in Florida, and found really high percentages of exposed turtles in populations that had zero tumor prevalence. So how did they become infected if none of the turtles have tumors? So this is a likely explanation for that. To follow up on that study, right now I'm doing a study on loggerhead and green sea turtles in North Carolina. Historically, FP has not been a problem for sea turtles in North Carolina. However, it's becoming more and more prevalent, either because the water is getting warmer or the sea turtles are transmitting it northward, or both. We're not sure, but we're just doing a survey of free-ranging turtles, working with NOAA to get samples from them, and also rehabilitating turtles at the Karen Beasley Rehab Facility to see if we can identify the prevalence of this virus in asymptomatic turtles or subclinical turtles. I'm doing another study. So in all these pictures, you may have realized, or you may have noticed, that the tumors can take lots of different types of appearances. They can be these flat plaques, they can be sessile and smooth, they can be papillomatous. This is the most common presentation in Florida. Or they can be polypoid and smooth like grapes. And in another study, they noticed that there are 4 different variants of this virus in Florida. There's A, where's B? B, C, and D. D's only been reported in loggerheads, and A, B, and C have been reported in greens. And so it got me to wondering, are these viral variants related to tumor morphology? So does the variant identity predict what the tumor looks like, and therefore its virulence within the host? So we're doing a study now with the Marathon Sea Turtle Hospital. We're looking at using molecular biology to look at virus variant identity in loggerheads and greens, again, and statistically evaluating those relationships between morphology or tumor morphology, species, and variant, and looking at both free-ranging and rehabilitating turtles and comparing the results. So just to summarize, overall, I think I tried to make it clear that wildlife rehabilitation has both benefits and drawbacks. It can have conservation benefits in cases of threatened or endangered populations, but the most important concept to me is that access to wildlife provides an excellent avenue for disease research and education. And I just want to thank all of my collaborators. I work with a lot of people, certainly could not have done any of this stuff alone, including the rehab facilities and University of Georgia and Harbor Branch. And I'll take any questions, and also this is my email address. If you have other questions or want to email me, have ideas you want to share, please feel free. So thank you very much.